prelude still? Was that it?
That was
Okay.
the prelude.
Sorry. I didn't recognize it. Good morning, everyone. We have an opening reading entitled Stem Cells by Amit Majmudar from 2013. In the hospital's hothouse, cardiac strawberries blink on a vine. A walnut shell hides a brain rich in good fat. A lychee's peel, a pale eyeball high in vitamin C. The doctor has good news. His pharmacopoeia has given way to a cornucopia, one that spills ovarian grapes and bananas that promise never to go soft. A single stem has birthed fruit that shall not be forbidden us. The pomegranate spleen, yay. The kidney bean kidney shall be ours. Splendid, delights a voice over the hospital PA system. Splendid, you summer sweet sons of Adam, using an apple seed of knowledge to grow the tree of life. Just a few very short announcements. And all this information is in the program that you've been given or that has been emailed to you. We do ask that you wear masks. We have an awful lot of vulnerable people who attend here, so we thank you for that. Second Tuesday discussion group will be September 13th. Not in the program, our topic is effective altruism. Still working on that. The board does need to know about how many people will want to visit the Hindu temple on October 30th, so please get that information together into Adrian in the office. Remember, please, to support and, if possible, to walk for the ALS walk on September 25th in honor of our former member, Leslie Edwards. Yesterday was a beautiful, crowded, joyful service memory for Debbie Palmisano. I'm sure she was here with us. I did speak with MJ Pearson this week. I think, depending on a doctor's report yesterday, she may be going home today. I'm sure she will still appreciate phone calls and cards. So give her a call if you do want to visit, or even if you just want to talk. She's feeling pretty good and welcomes the communication. So let us begin. I will begin by lighting the chalice.
Rise up, O flame, by thy light glowing, show to us beauty, vision, and joy. Rise up, O flame, by thy light glowing, show to us beauty, vision, and joy.
Thank you. This is a time to prepare our hearts and minds for what can be experienced this morning. Please feel free at this time to drop a prayer stone in the water bowl on the table here. The medicine wheel is outdoors. If you would like to walk outdoors and drop a stone there, you may, but I think that is a typo. Anyway. So let us sing together. Wake Now My Senses. It's number 298. And the lyrics are on the board.
Wake now my senses and hear the earth call. Feel the deep power of being in all. Keep with the web of creation your vow. Giving, receiving as love shows us how. Wake now my reason, reach out to the new. Join with each pilgrim who quests for the true. Honor the beauty and wisdom of time. Suffer thy limit and praise the sublime. Wake now, compassion, give heed to the cry. Voices of suffering fill the white sky. Take as your neighbor both stranger and friend, praying and striving their hardship to end. Wake now, my conscience, with justice thy guide. Join with all people whose rights are denied. Take not for granted a privileged place. God's love embraces the whole human race. Wake now, my vision of ministry clear. Brighten my pathway with radiance here. Mingle my calling with all who will share. Work toward a planet transformed by our care.
Thank you, Michael. At this time, please stand as you are willing and able and recite with me our unison affirmation. We pledge to create a welcoming community that celebrates human diversity, encourages spiritual growth, fosters responsiveness to our world, and commits to the future of all children. Thank you. We are fortunate today to continue our series about healthcare and our principles with an address by Vered Shacham Silverberg, who is a research associate at Cincinnati Children's Hospital Medical Center. And we welcome her, and afterwards, if you have questions or comments, she is happy to take those.
Thank you. Hello, everyone. Yeah, I'll just wait for the slides to come up, but in the meantime, I'll just introduce myself. So I'm Vera Chacham Silverberg. I came to Cincinnati almost 5 years ago, all the way from Israel, to conduct research at Cincinnati Children's. After doing my PhD in Israel, well, after living all my life in Israel, basically. And I'm currently a research associate at James Wells Lab at the Division of Developmental Biology, where we use stem cells and animal models to study the development of the gastrointestinal tract. So everything from our esophagus all the way down when the food comes out. So, today I would like to share with you a little bit about recent current advances in stem cell research. And I will focus on what is currently being done in research, how we are using stem cells to understand basic human biology, but also trying to develop new treatments for different diseases, and what is today the current status of it. Of course, I'm not a know-it-all, and I tried to bring as much as I can with the time we have. Hopefully I'll be able to go through everything. In the end, I'll be more than happy to answer your questions. Of course, if I'll be able to, I'll try. Yeah, so we can start. So just two definitions that are very important when we are talking about research. We have basic research versus applied research. So basic research is everything that is done that it's only driven by curiosity of researchers. So there is no money behind it or like huge pharmaceutical companies trying to make profit. It's just our human nature of curiosity, trying to understand how our human body works, how the nature works. So just, we are driven by the why. So why, what, how questions. And there is no bad question here. You can ask everything. It's from a very, very small question. How is this very specific gene in our body is working, to a much larger questions, for example, questions in ecology and climate change, so a much larger question. So everything is valid. When we're talking about applied research, is actually research that is more towards a solution for a specific problem. So for example, I would like to find a specific drug for this disease. And I'm a pharmaceutical company, well, I also want to help people, but there is also, of course, a financial power behind it that wants to make profit out of it, which is not a bad thing, because without profit, nothing will be done. But this is the main difference between the two of them. Next, please. And, just one foot. But we can see that they are highly connected. So basically, almost all of the applied research started in basic research. Almost all the drugs that are being used were first discovered, I would say, by accident, by some researchers studying a very specific biological mechanism and found this new target for a drug. And that drove him to connect with a pharmaceutical company to create this drug. So they are highly connected. One won't exist without the other, because money from pharmaceutical company also goes into academia and universities to pay for basic research as well. So they can't survive one without the others, and we need both of them to drive our understanding. Next. For many, many years, researchers used various different kinds of animal models to understand biology, and also human biology. Yes, we used the fruit fly to understand human biology, and the reason is we all share similarities. Our DNA shares some similarities with all of those various animal models that are here on those charts. None of them are perfect. Each of them have their advantage and disadvantage. Some of them can be used better to study, for example, embryonic development, because, for example, when you're using the zebrafish, the embryo is transparent, so without a microscope, you can see the entire development of the embryo in your eyes. So it's, of course, very helpful. Others are more expensive, so it's a disadvantage, but they are more similar to human, like rodents, mice, and rats, so of course it's more beneficial to us. But none of them are perfect. And none of them are perfect, especially when we want to study human disease, human development, and try to look for therapeutic solutions. And the reason is none of them are identical to us. So when we want to try to find a cure to cancer, but cancer is a little bit different in mice than in human, well, will the drug we use on mice will work the same on human? Of course, we need to adjust the dosage just because of the size of the animal, but it's not only that. It might work completely different. And this is where stem cells try to close this gap. Next. So what are stem cells? Hopefully you can see the arrows are very, very bright, so it's a little bit hard to see with the light, but stem cells are basically cells that are defined by having the capacity to self-renew, so they can make more stem cells, but they also have the ability to differentiate and to generate differentiated specialized cells, so every kind of cells in our body. So our skin cells are different from our neurons, our brain cells are different from our liver cells, are different from our blood cells. Stem cells can make all of them, and they can make more stem cells. Next. You can see that there are three types of what we called multipotent cells. So potent come from the word potential. The cells have potentials to make more than one type of cells. So we have totipotent. Toti comes from the whole. It can make the entire embryo, and when I'm talking about the embryo, is also the placenta, so also stuff that won't be in the baby, but are necessary for the embryo to grow inside the mother. Pluripotent are cells that can make every cells in our body. This is what we call, when we say embryonic stem cell, this is what we mean. So with embryonic stem cell, you can't make an entire embryo because you need the cells of the placenta, but you can make all the cells of the embryo separately, if you understand what I mean. And multipotents are cells that can make only a specific type of cells, several types of cells. So for example, our blood stem cells, which are called scientifically hematopoietic stem cell, can make all the different cells in our blood system, because our blood system have many types of cells. We have red blood cells, white blood cells. White blood cells have many, many different types. So hematopoietic stem cells are multipotent stem cells. They can only make blood cells, but they can make different kind of them. Next. There is another kind of stem cells that are called, it's actually the multipotent stem cells. They are also called adult stem cells. The reason they are called adult stem cells is because they are found in our body as adults. So embryonic stem cells are only found in the very early stages of embryonic development. So when the embryo just starts to develop, the first initial steps of the development are embryonic stem cell. But from a certain point, those cells start to differentiate, and then in each organ of our body, we will have what we call adult stem cells. So as I talked about hematopoietic stem cells, so hematopoietic stem cells reside in our bone marrow. But our skin has an adult stem cell, our intestine have, and so on. Some organs have higher regeneration capacity than other, which means they have more stem cells than other, so they can recover better after injury. So the mission of adult stem cells is when there is an injury, for example, to generate new tissue. And we can extract adult stem cells from tissue biopsies and grow them in the lab. Next. This is just two examples of adult stem cells. So on the left is the one I already talked about, the hematopoietic stem cell that resides in the bone marrow. As you can see here, the different type of cells that they can make. Different cells of the immune system, the white blood cells, red blood cells, T cells, and B cells. B cells are the cells that make antibodies, which is a very hot word in the last 2 years. But another example is intestinal stem cells. So the intestinal epithelium, which is the inner layer of the epithelium of the intestine, is changing every 4 to 5 days. The entire population of the cell there is changing every 4 to 5 days. And the reason for that is the huge amount of, the huge capacity of adult stem cell there. So if you look at the figure there, in the bottom area, what is called the crypt is the area where the stem cells reside, and they keep creating more and more stem cells. But also those stem cells starts to climb up and differentiate to the different types of cells of the intestine. So our intestine have different cells that have different jobs in the function of our intestine. And when they reach the top, they die and just shed into the lumen of the intestine, and they are just being washed away with everything we eat and drink, and there is new cells coming instead of them. So when we have injury in the intestine, it has a very high capacity of recovery. Next. So for many, many years, people thought there is only one direction that stem cells can go through to. Stem cells can become differentiated cells, but not the other way around. But in 2012, the Nobel Prize in Physiology and Medicine was awarded to two scientists that actually discovered you can take those differentiated cells and reprogram them back to stem cells. Those two scientists were John B. Gordon and Shinya Yamanaka. Next. So John B. Gordon is professor from the UK, And what he discovered is that if you take an egg cell, you take out the nucleus of the cell is where the entire DNA is. And instead of it, you put the nucleus of a completely differentiated cells, let's say a skin cell, you can create an entire animal. This is what we call cloning, and this is what basically gave us Dolly the sheep. This is how Dolly was made. They took a cell from an adult sheep. They took out the nucleus from it. They took an egg from a different sheep. They put the nucleus into the egg. They waited several cycles of splitting of the cells in the dish, and then they put it in the womb of an adult sheep. And she carried the pregnancy and gave birth to Dolly the sheep that was identical to the sheep that they took the skin cell from. So this is what we call cloning. The second winner of the Nobel Prize is Shinya Yamanaka. Next is a Japanese professor. And actually what he found is that you can take this skin cell, put him in a dish, and it doesn't have to be a skin cell, it can be any type of differentiated cells in your body. And you give them only 4 specific factors that are now known as the Yamanaka factors, and it will transition the cell into a pluripotent stem cell. So basically reprogramming the cell into a pluripotent stem cell. And he named these cells induced pluripotent stem cells because we are inducing the pluripotency in cells that were already differentiated. As I said, you can take skin cell, you can take every cell of the body, but the most used cells are skin cells and blood cells for the basic reason that they are the easiest to take from people without harming them. And to use them in research and in medicine, this is why this is the cells that are mostly used. Next. So now we can take a biopsy, let's say a skin biopsy. We give them these 4 Yamanaka factors that are actually listed here, but there is no reason for you to remember them. And they become stem cells. But now, from our knowledge of many, many years of study and development in animals, we can make those stem cells every different kind of cells in our body. So we can differentiate them to blood, we can differentiate them to skin, we can differentiate them to liver cells, to pancreas cells. So now we have the option to start with a skin cell and finish with a neuron. Start with a skin cell, finish with intestine cell, which allow us, if we think about the application of it, we can take, if we have someone that has a disease in his intestine, it's a genetic disease, this gene that is mutated is only important in the intestine. So if we'll take his skin cell, we can make them into induced pluripotent stem cells, then we will transition them into intestinal stem cells, and we can try and study his disease in the dish. We can try and see which drugs work on his cells specifically, and I will give many, many examples for that as we go on. Next. We can grow those cells, those new differentiated cells, in what we call 2D, just a monolayer of cells. But as we all know, our body is not 2D. Our organs are not 2D. Our organs are all 3D. They have a very specific organization. They have a very specific structure. And this structure is many times very, very important to how their function. So the next step was to make those cultures 3D cultures. And when we use very certain conditions in the lab, we can actually make those induced pluripotent stem cells to differentiate into 3D cultures. And we call those 3D cultures organoids. And it comes from basically mini organs. They have what is unique about organoids when you compare them to the 2D culture is that in an organoid, you will have all the different more than one kind of cells that you will find in an organ, because as I said, our intestine have many different types of cells. So when you will have a monolayer of cells, they will usually be all the same type of cells. But when you have a 3D cultures, you will have different kind of cells. You will have the same structures, even though it will be miniature, you will have the same structures you have in the intestine. So they are organized and function like the cells in the organs. They give you diversity, complexity, and function, which means we can use it for many more things. And you can see in the diagram on the right how we can make organoids for almost every organ of the body today. Next. We can also make organoids from adult stem cells that I told you about earlier. So instead of differentiating induced pluripotent stem cell to a specific fate, we can just take biopsies from patients, and those biopsies have the adult stem cells. And when we put those adult stem cells in a very specific condition, they will create those organoids. But they, of course, can only make organoids of the specific organ you took a biopsy from. So if you took a biopsy of the intestine, you can only make intestinal organoids. When you use induced pluripotent stem cell, you can make organoid of every organ of the body. You just need to know the protocol. You just need to know the recipe to make it. But with adult stem cell, you can make very specific organoids. Next. You can see here, and this is, I think you can already say it's already old. There are probably more organs that we can make now, organoids that we can make now. But you can see here the different organoids we can make from either embryonic induced pluripotent stem cell or from adult stem cell. So you can see that for some of them we can make from both. But we can, for some of them, for example, for the brain, we can make brain organoids only from induced pluripotent stem cells, also because you don't really take biopsies from the brain unless it's from like a tumor to study the tumor. Just for example, it says here that you can only make, now I don't see it. Well, the esophagus, yeah, here. The esophagus is marked as only adult stem cells, so I can tell you this is not true because I'm studying the esophagus, and I'm making it from induced pluripotent stem cell. So this is already old version of the diagram, but as research continue, researchers are able to make more and more organoids from more and more organs. Next. So what we can do with organoids? We can do screening of toxicity of drugs. So for example, we have 500 types of drugs, and we want to make sure that they don't cause damage to the tissue. So instead of using it on people, we will use them on organoids and see if there is any damage caused to the tissue. One of the main organs that is being damaged from drug is liver. So we can use liver organoids and try, even if the drug is not intended to treat anything to do with the liver, we just want to check if it's toxic for the liver. We can use them to study basic development. So as the organoid develop in the lab, in the dish, it basically follow the develop of the organ in the embryo, so we can follow development. We can intervene in specific steps and see what are the mechanisms that drive each steps. We can study genetic disease, see why specific mutations cause specific harms that we not yet understand, and so on and so forth. So there is basically a very high diverse usage of organoids. Next. So another very important tool that is being used today, and you probably all heard about, is what we call CRISPR-Cas9. And actually, CRISPR-Cas9 is a very good example of something that started in basic research and is now being applied for both basic and applied research, and it's a highly useful tool. So CRISPR-Cas9 was basically just discovered by being the immune system of bacteria. It was just researchers studying bacteria and found out they have this ability to fight viruses that attack them by cutting the DNA of the viruses. But the system was then modified by researchers so we can use it not only on viruses, but we can use it on humans, in mice, and in every other organism to modify the DNA. Next. And therefore, it was awarded the 2020 Nobel Prize in Chemistry for the development of a method for genome editing. It was awarded to two researchers, Professors Carpentier and Duedna. Carpentier, she is from Paris, and Duedna, she is from California. And both of them separately, but at the same time, were able to use the CRISPR-Cas9 technology to utilize it to change DNA. And this is basically being used all the time. So what it means is that I can take now a cell, a human cell, and change a very specific gene in it to create a very specific mutation or to correct a mutation and to study what is the effect of the change that I did. Next. So what we can do now, if we combine those two Nobel Prize technologies, induced pluripotent stem cell with CRISPR-Cas9, is that we can use it to cure human disease, because we can take cells from a patient that have a genetic mutation causing this disease. We can create induced pluripotent stem cells from it. We can correct his mutation, differentiate those cells to the cells in need in his body that are harmed by the mutation, and then, first of all, use them to study, but also put it back in the human. It was generated from him, so we are not afraid of any kind of rejection like we are scared of in organ transplant. But he will have these now cured cells in his body. So this is an amazing use of these two technologies. Next. So just to let you know that in the last couple of years, there is a huge effort at Cincinnati Children's to use stem cell and organoids to find cures and understand disease. This is called COSTUM, the Center for Stem Cell and Organoid Medicine. It's a multidisciplinary teams of scientists, clinicians, and entrepreneurs using advances in developmental biology and stem cell technology to revolutionize personalized medicine and improve patient care. It is led by these four researchers. The second from the left is my boss. And basically, what we are doing is using organoids and stem cells to study almost every organ in the body you can think of. The benefit of working at the children's hospital is that we have access to patients' biopsies, many of them with genetic conditions, so we can study, but we can also help them and understand their conditions and tell the physician what we found out so they can treat them better. So this effort is helping everyone. Next. So I just want to give you two examples of research done in Cincinnati Children's of using embryonic stem cell, induced pluripotent stem cells. So the first one, just a little bit introduction about cells. So this is basically finding possible treatments for patients suffering from severe malabsorptive diarrhea. It was found that in babies that it didn't matter, from the day they were born, it didn't matter what you fed them, they had malabsorbing diarrhea. Their body wasn't able to keep down nothing, not breast milk, not formula, not anything. And what was discovered is that there is a very specific, unique type of cells in our intestines. Those cells are called enteroendocrine cells. They only make 1% of the cells in our intestine, but they're crucial for nutrient absorption. And those patients had mutation in only one gene, the gene called neurogenin 3, and they didn't have any of their enteroendocrine cells, which means their body couldn't absorb anything. Also, they had diabetes from a very early age, because those cells are also very important in our pancreas. But a study done in our lab using intestinal organoids and using CRISPR to mutate this one specific genes to make organoids with the cells and compare them without the cells and compare them to organoids without them, was able to find that treating with one single hormone called PYY was able to restore their intestine function, which means injection of only one hormone daily might solve this problem. And of course, those kids, without treatment, you have to give them a GI tube to feed them. They can't, they have to get the food directly to their blood system. Anything can't go through their intestine. Some of the treatment was like surgery, which wasn't always very successful. So here we have maybe an easier solution. And this is currently being studied, and hopefully it will go on to become a treatment for those patients. Next. Yeah, so this is just showing the results of the study and how treatment. So if you look at the bottom left, you will see this is on mice that didn't have those cells. And in gray, you see that most of them die before they reach age of 2 months. But by treatment with this PYY, you can rescue them. And this is the red and purple lines compared to the black one, which is just normal animals that didn't have any problem. So basically, we can see how this treatment, daily injection of this one hormone, really improved the survival of those animals. As I said, those patients also have diabetes from a very early age, but the age of onset is different between different patients. Some of them will develop diabetes from basically infancy, so 3 months old, and others only when they are 12 or only when they are 20. Why? What is the difference? All of them don't have those cells. All of them have mutation in this neurogenin 3 gene. So what another researcher in our lab did is generated organoids with a different type of mutation. So each patient has a mutation in a different area of the gene. So if you can look in the diagram in this black and red thingy that is marked there, this is basically a diagram of the gene. And you can see the arrows marks different sites of mutation. And every site of mutation caused a different phenotype, a different onset of diabetes. And he was able, the graduate student that did this study was able to identify which area of the gene are important more for the diabetes and cause an earlier onset compared to others. So, and this helped us identify the differences between the disease onset and the severity. So this is another example of using both CRISPRs and induced pluripotent stem cell to study diseases. Next. Now I want to give you an example of the use of adult stem cells to find cure for diseases. The disease I'm going to talk about is cystic fibrosis. It's a devastating disease. It's caused by mutation in a single gene that code to a protein called CFTR. CFTR is cystic fibrosis transmembrane conductance regulator. They are always so easy, the names, and very, make sense. So this protein is on the membrane of cells and regulates secretion of sweat, digestive fluid, and mucus. by controlling the move of salt and water through the cell, through cell walls. And because of this specific role of this protein, patients with a mutation in this gene have buildup of thick, sticky mucus in the lungs, and this causes bacterial infection, coughing, and wheezing, and it can be fatal. Because it's important also for sweat secretion and digestive fluids secretion, they have also problems in other organs in our body, as you can see under A over there. But the life-threatening condition is the one in the lung. So they can live with like the other conditions, but if you won't treat the lung condition, it can be deadly because they won't be able to breathe, which we all know is quite important. Next. There are many different mutations. So as I said, even though all patients of cystic fibrosis have mutation in the same genes, they don't have the same mutations. In the US, 1 in 3,500 is born with CF. Only recently, drugs were developed to combat the underlying symptoms and improve patient quality of life. There are more than 2,000 known mutations. So in this one gene, you can have mutation in 2,000 different places in the gene, and you will have the same disease. But what it means is that you will respond differently to treatments. And finding the best medication for each patient can be costly and delay treatment. So one medication can work very good on one patient, but can be completely useless on another. And as you all know, in the US, healthcare is very expensive. To use a drug that is not working on someone, and then for it not to do anything, so you already paid a lot of money, and now you need to try a new drug. It's expensive, and it's problematic. You can see here just an example of different type of mutation and the different type of therapies that are used for each one of them. Next. We can take biopsies from those patients, and the easiest is to take an intestine biopsy. It's not as harmful as lung biopsies, and because this protein is also present on intestinal cells, we can also use intestinal organoids to study the function of the protein. And because, as I told you earlier, the intestine have a very high regeneration capability. When you take a biopsy, the intestine will heal itself very, very quickly, so there will be no damage to the patient. So you take the biopsies, and you generate organoids, and you use the organoids to test the different medication. Next. So basically, what some researchers in the Netherlands found is that when you treat those intestinal organoids with a very specific thing called forskolin, it's some kind of reagent, and healthy organoid will swell because water will come in. But a damaged one won't be able to get water into the organoid, and it will remain in the same size. So you can see on the right, under C, the increase of size of the dark blue, which is the normal healthy organoids, while the damaged organoids stay at the same size. So basically, we can study the difference between healthy and damaged organoids. So next. Okay, I'll finish about on the CF, and then I'll go to, even though I have many more to tell you, I'll just finish this story and then, okay, next. So basically what they found is that if you treat with drugs, you can see if it restores the healthy behavior of the damaged organoids. So now you can study the specific different drugs on those organoids and see which drugs is able to restore the damaged organoid activity, so make it swell. And basically, in the Netherlands, they were able to get reimbursed by insurance companies to treat those patients because they were able to find treatments for patients that they didn't know which drugs work for them. I don't know if it will ever happen in the US. I might not be as hopeful, but in the Netherlands, it improved the quality of life of many CF patients that were now being able to get the medication that worked best for them. Okay, so unfortunately, we don't have more time. I have a lot of other things to share with you, but I will happy to answer your questions.
Hello? Hello? I have a question.
Yes.
Can you hear me?
Yes.
I'm Johannes Bjorner, and we are in Mexico right now.
Oh, wow.
And there's a poor village where the children have world record in kidney disease because they drink water from a big polluted lake. And I'm just wondering whether stem cell therapy can help heal those children that are dying in large numbers because of their kidney disease.
Well, there are kidney organoids, and there are kidney organoids being studied in Cincinnati children. I don't know what is the specific disease they have, but I'm sure if it would have been known to specific researchers and they will be studied, hopefully it will be able to be treated. I'm not an expert on kidneys, so unfortunately I can't really tell you exactly what, but it is being used for studying kidney disease as well. So hopefully in the near future there will be a treatment for them.
Okay, very quickly, a second question. Can stem cells now be used to repair retina in people who have macular degeneration?
Actually, yes. It's actually one of the almost only organs that you can already treat with stem cells just because you have a very easy access to it. There are some treatments. Most of them are still under clinical trials, so they're not yet FDA approved, but they are being tried to treat retina conditions, so yes.
Thank you.
This may be the same question. Many of the older folks develop macular degeneration. So what's been done with stem cells to try to attack that problem?
So the short answer is, there is no short answer, but basically it's been used, first of all, to see if we can replace the damaged cells. Can we insert stem cells that were differentiated to new healthy cells? If we will put them in, you were talking about the retina as well?
Yeah.
So if you put them in the retina, will they replace the damaged cells, which is an important thing to check. If there is a genetic condition causing it, can we fix the genetic condition? Can we treat it with drugs? So we can try on the dish to treat the cells with drugs and see if we can do that. So there are various aspects of that. But the major treatment that is being trialed right now is injection of new healthy cells to see if they can replace the damaged cells and restore eyesight.
Okay. I just wanted to say, too, that if you really want to hear this in detail and get really get a lot of the information in a way that Dr. Silverberg has been teaching through Ollie.
Well, I only gave one talk. I don't know if they will request me to come back.
Well, we might request you to come back to Ollie, but anyhow, so she's been there, so keep your eye open, those of you who are Ollie attendees, and see if she's on the agenda.
Basically, I want to get some clarification. So let's say you took a disease like leukemia, and you created an organoid or something, and you injected it into the patient. So now you've just put a few cells in there. Is the goal then is for that little bit of cells that you just injected grow to the point, and the bad cancerous cells just die off? Is this a given?
Okay, so I'll divide my answers to two. The first part is regarding leukemia. So leukemia is actually blood cancer. The treatment for blood cancer is what usually bone marrow transplant. Basically, what bone marrow transplant means is that we kill the entire blood stem cells of the patient, and instead of it, we put new healthy stem cells that will hopefully make the entire new immune stem cells, blood cells of the patient. This is actually an amazing, very good example of stem cell treatment. It's basically the only approved stem cell treatment today, like the actually approved, FDA approved, that is highly used. I didn't get time to reach this part of my talk, but what I wanted to say is one of the main problems of organoids today is that they are still very, very small. And no, they aren't able. We do hope that when we reach a bigger size and we put them in the organ, they become part of the organ, part of the tissue, but they're still too small for that. One of the things we are doing to grow the organoids, and another problem with the organoids is that they are still very young. When I say young, I mean they are very embryonic-like. So when we look at intestinal organoids, they are more similar to intestine of a second trimester embryo and not an adult intestine. The reason is that at a certain point, they need more than what we can give them in a dish. And what we are trying to do to mature them in size and in function is to transplant them into animals that their immune system was changed to the human immune system, so they won't reject them. And then the animals provide blood vessels and other nutrients that are essential for the growth of the organoids. We are today doing that in mice and rats, but even mice and rats are not big enough. And one of the efforts, actually, by a researcher in Cincinnati Children is to use humanized pigs to transplant the organoids and get a much bigger size that will actually might be helpful for organs on demand, I would say. I would say that for some conditions, even a very small amount of cells can be curative, and a good example for it is diabetes, type 1 diabetes. In type 1 diabetes, what happens is that our immune system is attacking the cells that produce insulin in our body, beta cells that are in the pancreas, and it is believed that even introducing a small amount of beta cells will be enough to restore the production of insulin and be curative for those patients instead of relying on daily insulin injections. So this is an example where, and it was found that it actually doesn't really matter if you put those cells inside the pancreas. You might be able to put them in the liver and they will still do their function. So it means that for some conditions, we already have the tools, and what we have is enough. We just need to work further to make sure that it's healthy and we're not causing damage by introducing those cells into the body. But when we talk about organs on demand, we still have some way to go. So today, it is mostly used as a tool to find specific personalized medicines, like I told you about cystic fibrosis, or to find, to make sure that drugs are not toxic. So, and we also use organoids from cancer patients to see what kind of chemotherapy will work on this specific tumor but not work on this specific tumor. So it is widely used, but I agree with you, we are still a bit far from actually using them as replacement for organs. Thank you very much.
Thank you.
Thank you.
This is why science is really important. Thank you very, very much, Fahad. So I would like to move on. Michael, do you want to
Offering?
do some
Yeah.
Louis Armstrong?
Yeah,
Or
let's
go
do the
to the-
offering during it.
Okay.
Yeah.
So at this time is when we do ask you to contribute to our economic security at GNH. If you are a guest, please do not feel obligated to give. We are happy you are here. There is no need to give, but we will take the offering at this time. I have no basket.
I see trees green, red roses too. I watch them bloom for me and you, and I think to myself, what a wonderful world. I see skies white, clouds white, bright blessed day, dark sacred night, and I think to myself, what a wonderful world. The colors of the rainbow, so pretty in the sky, are also on the faces of people passing by. I see friends shaking hands, saying, how do you do? Really they're saying, I love you. I hear babies crying and I watch them grow. They'll learn much more than we'll ever know. And I think to myself, what a wonderful world. Yes, I think to myself, what a wonderful world.
Thank you, Michael. We do have a wonderful world. And as we learn more, it becomes more apparent. So we thank you very much. On behalf of our congregation, I thank you for your offerings today. At this time, we have an opportunity to express or share joys or sorrows, things that we are dealing with, things we would like to celebrate with one another. Excuse me. If you have something that you would like to share or ask for some support for, please raise your hand and we will get the microphone to you again. Thank you, Tim.
Just wish to share that yesterday was Richard's 83rd birthday.
Woo.
Thank you.
Congratulations, Richard. Happy birthday.
No more happiness?
Always good to share our growth. Thank you, Tim.
Oh.
Oops, over here.
A guest?
Yes, you knew I was a guest. Just because I don't look familiar. No, you're right, but this is my first time. And I attended Andy Rotterman's memorial service last Saturday. And I was just so impressed with the environment, so I went online and read about the congregation. So I just wanted to say thank you all for being here and for providing some place where I feel like I can finally belong after searching for 77 years.
Welcome back.
Well, thank you and welcome, and we hope to see you again. Wonderful. Okay, let us proceed.
Hey, Sue,
Yes,
on Zoom there.
I'm sorry.
It says, please grant my wife and myself peace and patience in raising a puppy and a 6-year-old. From Joel Ruggiero.
Joel, thank you. I apologize. I have ignored our people who are with us from afar. Sarah, is there anyone else? Thank you. And again, I apologize. Goodness. All right. Let us proceed at this time to our closing hymn. And we will sing together, Turn the World Around. Nice, zippy tune to send us forth for the week.
We come from the fire, living in the fire. Go back to the fire, turn the world around. We come from the water, living in the water. Go back to the water, turn the world around. We come from the mountain, living on the mountain. Go back to the mountain, turn the world around. Whoa, so is life. Ah, so is life. Whoa, so is life. Ah, so is life. Do you know who I am? Do I know who you are? See we one another clearly. Do we know who we are? Do you know who I am? Do I know who you are? See we one another clearly. Do we know who we are? Whoa, so is life. Ah, so is life. Whoa, so is life. Ah, so is life. Water make the river, river wash the mountain. Fire make the sunlight turn the world around. Heart is of the river, body is the mountain. Spirit is the sunlight, turn the world around. We are of the Spirit, truly of the Spirit. Only can the Spirit turn the world around. Whoa, so is life, so is life. Whoa, so is life, so is life. Whoa, so is life, so is life. Whoa, so is life, so is life.
Thank you, Michael, for that up-tempo version. Whew. I would like to, before our closing words, there's a prayer poem that was found to recite or to read during this particular service. It's called Stem C by Tyson Anderson. This is my body, which is given for you, but I'm not great. I have neither wealth, nor fame, nor grace. I cannot comfort with words, nor inspire to march. I am small and simple, so leave me this. Let me heal you. This is my body, which is given for you. Take this in remembrance of me. And so let us close the service and go on to a week with a new respect and a new understanding for the science that is happening all around us, here in Cincinnati and everywhere. The amount of research that is taking place to bring us healing and longer lives. A politician by the name of Jim Ramstad said, embryonic stem cell research will prolong life, improve life, and give hope for life to millions of people. So we thank you for being here. Let us conclude and go have a snack. Please join us in the Quimby room. Thank you.